Good afternoon, and welcome to another webinar sponsored by the North American Vascular Biology Organization. I'm Vinicio de Jesus Perez from Stanford University Medical Center and a member of the NAVBO Education Committee. I will be moderating today's session. We are pleased to welcome our speaker, Dr. Yuan. She's an assistant professor of pediatrics at Boston Children's Hospital of Harvard Medical School. She will present research entitled Mural Cell SDF1 Signaling is Associated with the Pathogenesis of Pulmonary Arterial Hypertension. At this time, I would also like to welcome Dr. Hong Shen, an associate professor also at Boston Children's Hospital. She will monitor today's questions. We handle questions in two ways. Throughout the presentation, you can type your questions into the question box in the control panel. These questions will be answered at the end of the presentation. Dr. Shen will compile and then post the questions to Dr. Yuan at the end of the presentation. At the end of the question and answer period, provided there is time, attendees will be able to ask any additional questions live in real time. If you have a question, please raise your hand by clicking the hand icon on the left side of your control panel. You will be recognized, your mic will be unmuted, and you will then be able to ask your question live. If you experience technical problems, please click the Help tab at the top of the control panel, then scroll to the bottom of the help screen for the GoToWebinar technical support phone number. This webinar is being recorded and archived on the NAVBO website for future use. Dr. Yuan is currently an assistant professor in the Department of Pediatrics at Boston Children's Hospital. She received PhD in biology from Temple University, Pennsylvania, and was then a postdoctoral fellow in the Division of Pulmonary Critical Care Medicine at Stanford University. Over her training years, Kay has been recognized nationally as a Parker B. Francis Fellow, a Fellow of the American Heart Association, and a recipient of the American Heart Association Scientist Development Grant, the Kurman and Comroe Young Investigator Award, and the American Thoracic Society Aldrich Research Award for Young Investigators. She is a cellular biologist, and her research focuses on the role of parasites in the pathogenesis of pulmonary arterial hypertension. Today, Dr. Yuan's presentation addresses how pulmonary artery smooth muscle cells and parasites are NG2 positive mural cells, and how they provide structural support to pulmonary arteries and capillaries. Please welcome Dr. Yuan.
Thank you, Vinicio, for your kind introduction, and thank you, NAVBO Educational Committee, for giving me the opportunity to present our work. So today I will talk about mural cell SDF1 signaling is associated with the pathogenesis of pulmonary arterial hypertension. And I will divide my talk into two parts. First, the introduction of mural cells, or parasite versus smooth muscle cells. Secondly, trace mural NG2 positive cells in hypoxia-induced pulmonary hypertension. What is pulmonary arterial hypertension? It is a devastating disorder which is associated with high pressure in the lung. If untreated, leads to right heart failure and death. The clinical presentations are cardiomegaly, attenuated split S2, dyspnea, renal insufficiency, hepatomegaly, ascites, and edema. The diagnoses are based on the X-ray of enlarged pulmonary artery, cardiomegaly, and enlarged right ventricle with the systemic pressure higher than 40 millimeter mercury. The diagnosis will based on the measurements from right heart catheterization with the mean pulmonary arterial pressure higher than 25 millimeter mercury, pulmonary arterial wedge pressure less than 15 millimeter mercury, and pulmonary vascular resistance higher than 3 Wood unit. So currently therapies are mainly oral and intravenous vasodilators with only the vasodilator properties fail to prevent disease progression or prevent the disease progression of leaving the lung transplantation. With its high mortality and morbidity, the only choice for patients with end-stage disease. So there's always unmet clinical need for new therapies. So let's look at the pathological features of the PAH. So here is a contrast image of a healthy lung vasculature. You can see the lung vasculature is like a tree with many, many branches. And when we look at the magnified distal portion, here's the pulmonary arterioles sending through and divided into small capillaries surrounding around the alveolar air sac. When we look at the cross-section of pulmonary artery, there's endothelial cells in the inner layer and smooth muscle cell and stroma cells surrounding it. However, when we look at the structure of the capillary, it's very different compared to the arterial, where there's no longer smooth muscle cells, but pericytes. Here is the contrast image of a PH lung. You can see the distal portion of this vasculature disappear, leaving behind only large-sized vessels. And the schematic image here showing the abnormalities throughout the lung pulmonary circulation. There is a disappearance of the small precapillary arterioles and their different degree of the muscularization throughout the arterioles. And here we're showing the H&E staining of a new intima on the left. And you can see the blood vessel luma minimized and surrounded by different kinds of cells. And on the right, that's a plaque formulation with disorganized endothelial cell channels. Therefore, the blood can go through and with accumulated blood pressure. So in order to form functional vessel, one of the key components is pericytes. So what are pericytes? Pericytes are mesenchymal-derived mural cells that wrap around endothelial cells of capillaries throughout the body. They are embedded with the basement membrane, which direct contact with endothelial cells, and its presence required for vessel stabilization, maturation, and remodeling. So far, according to our experience, the only known pericyte only find in the capillaries with a vessel diameter around 10 micron size. And the rest part of the location of the vessels, they are no longer pericyte, they are smooth muscle-like. Loss or reduction of vascular pericyte coverage associated with numerous pathologies, such as cancer, Alzheimer's disease, diabetic retinopathy, and their roles in pulmonary vasculature is under investigated. So right now, our lab and others have 3 distinct methods to distinguish them from other mural cell types. First of all, they should reside on capillaries with 10 micron size diameter, which I showed in your previous slide. Secondly, pericytes should have an oval cell body with punctate lung processes. Lastly, they are positive for NG2, PTGF receptor beta antibody staining. Here I'm showing a C56 black 6 mouse retina staining, and the NG2 is labeled in white and used to recognizing pericyte. And you can see it's typical oval cell body and the covering around the endothelium, which is in the green color, and the smooth muscle actin are used for staining smooth muscle cells. So there is another way, which is indirect labeling, by utilizing Krelok's-P system to visualize and label pericyte using a NG2 or PTGF receptor beta promoter-driven reporter mural line. Here is an example which was published most recently, showing the pericyte loss will lead to the neuron loss in the brain. And the authors use double labeling system, which here they label parasite in TD tomato color. And you can see its oval cell body covering around the CD31 endothelium in blue, and the smooth muscle actin used for staining for the smooth muscle cells. So using the same methods, we use a NG2 CRE-ER line label using TD tomato, and that's the first time we could see these cells in the mouse lung. And let me show you by this confocal reconstruct image. And here is a typical shape of a parasite with its lung processes covering 4 alveoli capillaries, and the endothelial cells are labeled with V cadherin. Let me show you one more time. Here, the white is V cadherin, and that's a lumen of a vessel, and that's a typical shape of a parasite. Again, we want to double confirm the identity of these TD tomato cells. We use SPC to label alveoli type 2 cells. You can see on this confocal imaging, there are 2 distinct cell populations. And when we zoom in this area, you can see the TD tomato cells with long processes covering around the capillary and very adjacent to this type 2 cells. Again, using confocal, we could visualizing or reconstruct the 3D structure and how these 2 types of cells interact with each other closely. And that's a zoom in, and that's a typical cell shape of a parasite. That's the green stain for type 2 cells. Well, then we want to study human lung parasite. Therefore, we isolating parasites from human transplanted lung tissue through PHBI network. So if I didn't specify all the samples, human sample we use in this presentation are from type 2, group 1 PAH, idiopathic PAH samples. Then after tissue digestion by collagenase, we use 3G5 IgM coded magnetic beads to sort these cells. And you can see their morphology on the matrigel and on the culture plate. Then we use FACS sorting to validate their cell identity, and they're positive for the following cell markers, and they're negative for smooth muscle myosin heavy chain, which is the mature smooth muscle cells, CD31, which is endothelial cell marker, and CD45, which is blood cell marker. So as its fundamental role in angiogenesis, parasites promote vascular stability. So if there's no angiogenesis, parasites are quiescent. If angiogenesis occurs, endothelial cells will secrete different kinds of growth factors and cytokines to recruit parasites, including PDGF and TGF-beta. And during my postdoctoral training, we identified Wnt5a as one of these growth factors which will recruit parasites. And once endothelials and parasite contacts are formed, notch afferent pathways are activated. At the meantime, parasites release endothelial survival differentiation factors to stabilize stabilize and form a functional vessel. So in order to study endothelial and parasite cell-cell interactions, we see them together on a matrigel. So here you can see the endothelial cells, they could form nice 2D tubes on a matrigel by themselves. Then when we incubate it with a healthy parasite, which enhanced tube formation and formed robust tubes, and when using PKH membrane staining, parasite in red and endothelial cells in green, we could see healthy cells are wrapping around this vessel. However, the PAH parasite failed to do so. They didn't enhance the tube formation, and they stay on the nodes. Interestingly enough, when we culture the PAH endothelial cells on matrigel, they fail to form nice tubes. And when we incubate them with healthy parasites, healthy one fail to enhance the tube formation. Here is the quantification of the number of tubes and number of networks, indicating the mutual communication between these cells, between those two cells are critical to form function vessel. And during the normal culture, I also noticed the disease PAH parasite pretend to proliferate faster, and this is further confirmed by the MTS stain assay, and they have faster cell cycle progression, and they have higher glycolysis measured by seahorse assay, showing they have higher metabolism rate. And when we mix the disease cells, PAH cells with in the collagen, these cells are more contractile, therefore formed smaller sized collagen gel. And lastly, that's the most recent data we obtained by applying RNA-seq analysis on healthy parasites, disease parasites versus healthy smooth muscle cells and disease smooth muscle cells. And by hierarchically clustering analysis, we see the disease parasites share more similarities with the smooth muscle cells. And next, we want to see the parasites and smooth muscle cells on patient tissue. Here I'm showing a control slides where the mature smooth muscle cells are indicated in red as by the smooth muscle heavy chain, and CD31 standing for endothelium in green. And the NG2 you can see indicated by the white arrow here, the NG2 showing these cells are on the capillary. And on this occluded pH sample, you see the smooth muscle cells are surrounding this occluded lumen, and the right arrows showing the positive NG2 staining, indicating these occluded vessels may be a mixture of smooth muscle cells or parasites. So far, I've been showing you a major pathological hallmark of PAH. It's combined of small vessel loss, and also muscularization of microvessels, maybe due to the PAH parasites, and therefore resulting in vessel progressive narrowing and occlusion. And the RNA-seq analysis demonstrate similar gene profiles in PAH parasites versus pulmonary arterial smooth muscle cells. So let me switch gear and introduce you to the second part of my talk, which will be trace Mural NG2 positive cells in hypoxia-induced pulmonary hypertension. So this is a slide I showed you before, and there's different degree of muscularization in the different size of arterioles. And in the previous published literature, parasites are known to be as multipotent stem cell-like. That means under different stimulations, they can become fibroblast, myofibroblast, adipocytes, osteoblast, macrophage, and one of the interesting lineage will be vascular smooth muscle cells. Back in 1980s, there's interesting and important publication reveals the ultrastructural parasite in lung biopsy materials from children with congenital heart defects. And this is the very first publication showing and indicating parasite could be a precursor of smooth muscle cells. And most recently, there are publications showing parasite can differentiate into muscle fibers, and parasite are progenitors for coronary artery smooth muscle. Therefore, my hypothesis is parasite gain smooth muscle-like phenotype in pathogenesis of pulmonary arterial hypertension. So in order to study this NG2 positive cell lineage, we use and leverage the Curie-LOXP system by using the NG2 Curie-ER line crossed with TD tomato flux line. And NG2 Curie-ER line have been used to selectively label parasites in neurobiology research, especially in blood-brain barrier studies. And after tamoxifen treatment, we could selective label parasites and NG2 positive parasite in smooth muscle cells in the TD tomato color. So experimental design is we will subject the mouse to hypoxia treatment, which is a method we induce high pressure in the lung in pH animal studies. Then we will do lineage staining and cell marker staining. And in the second aim, if we see differences, we could sort out these cells and subject to RNA-seq analysis to understanding the gene expression pattern. So here I'm showing you the landscape of a tamoxifen-treated NG2 TD tomato lung. By carefully dosing tamoxifen, the majority of TD tomato positive cells were found in the parenchyma and the capillary. However, we do find positive TD tomato recombination in vessel size larger than 100 micron, as we see here in this area here, suggesting the non-specific labeling and leakage of this mouse line in the smooth muscle cells. However, with the precision and the minimum tamoxifen dose per body weight, we are able to consistently label the same amount of TD tomato cells in capillary. And we also notice there's no TD tomato positive cells between vessel size 10 micron to 100 micron. Therefore, in the rest of staining, we are extremely careful with the size of vessels and only choose the vessel size smaller than 20 micron size. And here I'm using the antibody staining to double confirm the cell identity. So for the parasites, which we use standard NG2 PTGF receptor beta staining, and for smooth muscle cells, we use smooth muscle actin and smooth muscle myosin heavy chain. As a classical way to identify parasites, we use electron microscopy. As this is a collaboration with Stanford Microscope Core, and we applied array tomography to firstly label TD tomato positive cells, and use the same scale and alignment. We could use EM to scan the structure of that same cells. Here, I'm showing the same area as indicated in the dash box. And here is the labeling of the structure. BV represents blood vessels, and the AL represents alveolar airspace, and WBC is white blood cells. And you can see here is a longitudinal structure of a capillary, and here is the TD tomato positive cells. When we zoom in, we can see the TD tomato cells share the same basement membrane with endothelial cell, further suggesting this could be a parasite. Next, we want to look at these cells under hypoxia treatment. Here is the normoxide control mice. We can see these TD tomato cells in the parenchyma and the smooth muscle actin recognizing smooth muscle cells. And these TD tomato cells are 100% positive for NG2 antibody staining. And they are also positive for PTGF receptor beta staining. However, they are negative for myosin smooth muscle cell staining, indicating they are not smooth muscle cells. And again, using confocal, we can appreciate the 3D structure of this blood vessel. Here is the lumen, and the TD tomato cells are in the parenchyma, and they are negative for the myosin heavy chain expression. Very interestingly, after 21 days of hypoxia exposure, on this arteriole, we see the TD tomato cells covering around this vessel about the size 20 micron, and they are no longer Ng2 positive. However, they are still PDGF receptor beta positive, and now they are also become myosin heavy chain positive. Here is a reconstruction of these 3D vessels, and here is the lumen of the vessel, and we can see these TD tomato positive cells cover around this 20 micron size arteriole. And when we closely look at the parasite and endothelial cell interactions, here is the maximum projection of TD tomato covered arterioles. And when we look through each slice of these vessels, we could see the red layer is distinct from the green layer by different slice, indicating these TD tomato cells do not co-express CD31. They are covering outside the endothelial layer. And because we are interested in parasite and endothelial cell-cell interactions, so we want to look at the capillaries. Here is the 3D structure I showed you before. And on this hypoxia treatment, again, this is the TD tomato cells covering the arterioles. And we also found there's a reduced capillary density. This is further confirmed by using this advanced blue staining. Here is a normoxia mice, and after 3 weeks hypoxia, then we inject the advanced blue through the tail vein, and we could see the advanced blue binding with the albumin. That's why we see those blue spots on the lung. And by extravasitated the advanced blue from the tissue, we could see hypoxia mouse have increased advanced blue in density. And next, we use ficilactin to stain the whole endothelium and inject the 50 nanometer microbeads, which is also in red fluorescence. So if the vasculature is normal after the PBS flush, the beads will be washed away. And if there's leakage happen and loose junction endothelium, these beads will remain on the interstitial space. And here you can see a very nice and preserved lung vasculature. However, in this hypoxia lung, we see the red fluorescence beads remain in the vasculature, further indicating if these TD tomato cells left the capillaries may make these vessel loss and also the loose endothelial barrier function. So the model I proposed so far is after 21 days hypoxia, we see increased TD tomato coverage in the larger size arterioles. So based on the observations we see from the staining, we move to our aim 2 is fact sort these TD tomato positive cells. Then we want to understand if there's any altered gene expression level could explain the biological phenomena we observed. So using fact sorting, we can successfully sort TD tomato cells from Ng2 TD tomato line. Here is the heat map showing the 21 days hypoxia versus normoxia, and the PCA plot showing the distribution of the samples. And we found 296 significant altered genes. And by the strain analysis and GO pathway analysis, the top hit is cell migration, which is fitting what we observed in the staining I showed before. And in the middle of the strain analysis of this protein-protein networks, one of the target is CXCL12 catch our attention. So CXCL12 is the gene's name, the protein's name is SDF1. So it's an important molecule for developmental processes, including hepatopoiesis, cardiogenesis, vascular formation, neurogenesis, and maintenance of tissue stem cells. And their presence are required for oxygen tension, inflammation, extracellular matrix, mechanical forces can also alter its expression. So in the published papers, CXCL12 have been shown to be important cytokine to recruit parasite, and inhibition of its CXCL12 and its receptor CXCL4 access attenuate neonates murine in hypoxia-induced pulmonary hypertension. And elevated plasma CXCL12 is associated with a poor prognosis in the PAH. And we also look back to the RNA-seq data we had in the previous slides. And HP represents healthy parasites, PP represents PH parasites, and according to the different transcriptional analysis, we found, and also indicating in this volcano plot, SDF1 is one of the upregulated genes. And we also have a brief idea to compare this human data with murine TD tomato positive cell RNA-seq data from hypoxia versus normoxia. So we want to see if there's any common altered genes. And indeed, this table summarizes the commonly altered gene that affect in both human and mouse RNA-seq data. And CXCL12 is the top hit. And I think the rest of the common altered gene are also equivalent interesting. And then we want to confirm this SDF1 expression from the bioinformatic analysis in our samples. So we first stain this SDF1 in the NG2TD tomato mouse line. I'm showing you here, this is a normoxia mouse line, and here is smooth muscle actin recognizing a vessel and TD tomato in the parenchyma, and we see a minimum SDF1 expression in normoxia. However, in this hypoxia mice, we see a globally increased SDF1 expression in the parenchyma. Then we go back to the human sample, which we use the Western blot to measure the SDF1 expression in the 3pH patient samples and healthy parasite sample, which was used for RNA-seq analysis. We see there's increased SDF1 protein level as well as mRNA level. And I have staining here showing the PH parasite have increased SDF1 expression. And also we go back to the PH patient tissue samples. So we see increased SDF1 expression in the PH sample. And by collaborating with one of our fellows in our department, we measured the SDF1 expression level in the plasma sample, and we also found that they are significantly increased in patients. So next, we want to explore more of this SDF1 expression. We use the cells on the collagen gel. This is the data I showed you before, which the healthy cells mixed with the collagen gel after 24-hour incubation. The disease cells is more contractile, therefore forming smaller gel size. And with the healthy cells treated with recombinant SDF1, the healthy cell become more contractile and forming also smaller size gel. And with disease cells, we use sRNA to knock out SDF1. You can see once they knock out SDF1, the the disease cells are less contractile. Here is a quantification of the gel area. Then here is we want to treat the healthy cell with recombinant SDF1 to see if they start to express smooth muscle cell marker. And here is showing the healthy cells with IF staining. They're negative for smooth muscle actin, calpain, and smooth muscle 22 alpha. And with recombinant SDF1 stimulation, these cells starting to express these markers, indicating they may become smooth muscle-like. Next, we want to study the endothelial and pericyte cell-cell interaction under the SDF1 treatment. Here again is the matrix gel. By endothelial cells themselves, they could form tubes, and with the healthy pericyte stain in the red, they could wrap around the tissue as indicated in the yellow arrow. And with recombinant SDF1, this cell tend to stay on the nodes and not wrap around the tube. And with the disease cells, they fail to enhance the tube formation. However, with the SDF1 knockout RNA, siRNA, these cells tend to be rescued. And here is the quantification of total tube length, total branching points, and the covered area for this matrix gel study. So based on the cell study, we want to learn if the SDF1 have its protective role in vivo. Therefore, we use the NG2 clear line and cross with the SDF1 flux line and subject the mouse into 21 days hypoxia treatment. And you can see in this graph here is with SDF1 knockout in NG2 expression cells, we see reduced systemic RVSP systemic pressure compared to the hypoxia alone. And the Fulton index is used to measure the RV hypertrophy. And we could see these cells have no longer have RV hypertrophy. Then we also measure and the muscularized area in this arteriole. We could see these knockout mouse no longer have no differences compared to its control and have reduced muscularized percentage of these arterioles compared to the 3 weeks hypoxia wild type. So this is double confirmed with vibraton staining, and I'm showing here is a normoxia knockout mouse. They have no differences compared to the normoxia control, and the smooth muscle actin recognizing a big size arteriole. And for here with hypoxia wild type mouse, which we see smooth muscle actin are positive in the vessel size less than 20 micron size. This is consistent with the findings by Dr. Dan Grives' group at Yale University. And in our NG2 knockout, SDF1 knockout mouse, we see small positive staining on this bigger size arteriole, but no longer in these small size arterioles, suggesting reduced muscularized arterioles. Then this is the translational part of this research is we want to use SDF1 receptor inhibitor called CXCR4 specific antagonist, which is AMD 3100. So we use NG2 TG tomato mouse, and then we subject this mouse line with 3 weeks hypoxia and also AMD 3100 in a mini pump, and it lasts for 21 days. And after 21 days, we measure the systemic pressure. We see these mouse have reduced pressure compared to the PBS control, and they also have no reduced or RV hypertrophy compared to the hypoxia PBS control. And we also want to look at the TT tomato coverage on the arterioles. Here is the control mouse with AMD 3100 in the NG2 TT tomato mouse. We see this is just as the control I showed you before. And with 3 weeks plus PBS, we see the TT tomato cover the arterioles. And however, with AMD 31 treatment, we don't see this and reduced TT tomato coverage on the arterioles, and also reduced smooth muscle actin coverage on the small vessels less than 20 micron size. So far have been talking about pulmonary arterial smooth muscle cells and pericytes, NG2 positive Mural cells that provide structural support and coordinate vascular repair following injury. And pulmonary arterial smooth muscle cells and pericytes may share a common molecular mechanism responsible for their abdominal behavior in PAH. And we have identified SDF1 as a key player in driving NG2 positive Mural cells to precipitate vascular remodeling and in trigger pathological PAH pericyte activity. And in the future, we want to study SDF1 transcriptional factor HIF2A in regulating pericyte lineage during vascular remodeling in vitro and in vivo. And we also want to leverage bulk RNA-seq and single-cell RNA-seq to understand the genetic landscape that regulating the vital function of pericyte during vascular remodeling. And then lastly, as our long-term goal is to target pericyte as a potential therapeutic strategy. With that, that's the end of my talk, and I would like to give my sincere thanks to my colleagues and collaborators at Stanford, especially to Dr. Vinicio de Sousa Cruz, Mark Nichols, Joseph Wu, and Malin Rabinovich, and Christy Redhorse, and also other collaborators at Yale University, Dr. Dan Greif, and my new collaborators in Boston Children's Hospital, and also there's fundings to make this work happen, and also the strong support from my family and my 2 little precious. And so my the references and the protocols and about my lab and new recruitments, you can visit my website at www.yuankelab.com. And thank you for your attention, and I will happy to take questions.
Thank you, Dr. Yuan. I see that we may have some questions from the audience, so I'm going to turn this over to Dr. Chen so that these questions can be directed to Dr. Yuan.
Sure, thank you.
Okay, so may I ask the first question?
Of course.
Okay, so a beautiful presentation, and so the question could be not a kind of philosophical question. So that so do you know why the coverage of the parasites in the lung, the capillary is so few? You know, when you show this beautiful 3D images, so it's a very like a spotted cut, you know, like coverage, like just strategic coverage. So do you know why is that the case? And I have a second question, but you want to address this question first?
Sure. It's a very good question. So this is the question related to the NG2 CRIER line and TT tomato Mural model we used. So so there's as we know, there's no unique parasite markers. So far, the people use for study parasites are using NG2 CRIER or PDGF receptor beta CRIER. But because the non-specificity, how much you can use the tamoxifen to positive label these promoter-driven cells are still problematic. So therefore, we also come across the same issue, and we try to use the optimized tamoxifen dosage. Therefore, there's minimum smooth muscle cells could be light up in the vessel, and we have been optimizing these methods. So I will say about 40% of the TD tomato positive cells in capillaries are also NG2 positive. So these, and according to their morphology analysis, these cells, they are parasite looking. And therefore, after using the same dosage treatment, we put in these mouse in hypoxia. Therefore, we should proportional coverage on the arterioles. So if you use too much tamoxifen, they may, in the baseline, you will see more parasite and labeling. But at the meantime, you will get a lot of non-specific labeling of smooth muscle cells in the larger size arteriole. So in order to reduce this leakage and the non-specificity, therefore we just using the optimized dosage. And that's why you see a partial coverage. And also by other groups of people's research, so especially the contribution from Dr. Dan Grives' group, they've been showing the precursor smooth muscle cells also contributing to the muscularization of these arterioles. So my work is complementary to Dr. Grives' finding. I think it's not only the precursor smooth muscle cell contributing to the coverage, the parasite could also contributing to the muscularized vessel. So I hope I answer this question and make the answers that make sense to you.
Sure. Yeah. So recent publication by Taya McKinnon using the TD tomato. Actually, she described that without tamoxifen, that the TD tomato has a basal signal. So because the piece of between the two locks P, it's too short, 0.7 kb. So that has a basal level. So I understand your frustration with this line. The better line is using the MT, so you can convert the green cells to red cells. Yeah. So that's one thing. So okay. So I have a question from audience. I should prioritize that question first over mine. Okay. So then a question from audience from Dr. Xiaowei Gu. From Andin Clever's lab. So he has a question that is saying in culture the parasites are proliferating and they express NG2, but not alpha smooth muscle actin. When they stop proliferating and after they differentiating, they are differentiated. They actually upregulate this alpha smooth muscle actin expression. So his question is, could SDF1 inhibits the proliferation of your parasites and induce the differentiation? That's the first question. And the second question is that have you measured the EDU incorporation in both the in vitro and in vivo in these parasites are isolated from the patients?
Both are very good questions.
So,
I'm sorry.
Go ahead, yeah. So maybe you can answer first question first.
Yeah. So before I answer Xiaowei Wu's question, I would like to follow up the comments you just said about the MTMG line. So I indeed worked with, I tried this NG2 Korea line with the MTMG line, but the recombination was really low, first of all, and I found abundant smooth muscle cells labeled in bigger size arteriole instead of cells in the capillaries. So I don't think MTMG could be used to label, or its line could be used for NG2 Korea line. Just have this follow-up comment. And go back to Xiaowei's question. So first question is about SDF1 inhibition proliferation and induced differentiation. So in the preliminary data we tested, so the SDF1 is enhance the proliferation for my control cells, which I didn't show here, but it's enhanced the proliferation. And in terms of the differentiation, we just simply put on recombinant SDF1 on the cell culture media, and we measure the smooth muscle, alpha smooth muscle actin expression by IF staining. So this is also a work in progress, and we want to use CRISPR technology to make a stable cell line in order to answer firmly about the differentiation. But the proliferation, we do see it is very robust proliferation. And to the second question, which is the EDU, I did the apoptosis assay on caspase 3 and 7 measurement, and we see these PAH parasites are less apoptotic. And for the EDU measurement, I indeed tried on the NG2 titty tomato mouse line. And very interestingly, we found is these titty tomato cells are negative for EDU, indicating they are not proliferating. So we are trying to figure out why, and maybe something happened before, in between this hypoxia 0 to 21 days, and maybe something happened even in a very early stage of this hypoxia stage. So it's a work in progress.
Okay, good, thank you. So I don't see any more questions from audience. So just so will you elaborate that the comments you made, that saying that the MTMG line, so it's all NG2 Cree, right? So if we think about it, so why would these two lines differ so much? I mean, and then I was, and then giving you comments that you see from the MGR, the TB tomato mice, these hypoxia treated parasites are not proliferating. So that is sort of thinking that maybe the mouse model that you use and may not recapitulate what is that, especially this MGR TD tomato mice may not be the perfect model that recapitulate the human PAH.
Well, I think I totally agree. I think there's no standard line, Cree line we could label parasites in the mouse. This is because there's no unique cell marker. So in the MTMG line recombination with Cree ER line, I think it's because the way how the MTMG reconstruct is very distinct from TB tomato. The TB tomato is very easy recombination. For example, for the MTMG, I have to give up to 8 milligram tamoxifen in order to see efficient labeling. However, for the TB tomato, about 2 to 4 milligram of tamoxifen, you can see a very robust labeling. And you have to reduce the tamoxifen dosage in order to reduce the non-specific labeling of smooth muscle cells. And the best way to distinguish the model I described is to use clonal analysis, which is could be used the NG2 Cree ER line combined with brain bone mice and with a minimum dosing of tamoxifen. Then you can label single level cells, then subject mice to the hypoxia to see if the single cell could forming colony, a clonal expansion of single cells with the same color. So that's the ultimate goal to tell or distinguish whether this phenomenon we saw in the capillaries are solely coming from parasite, not from the bigger size arteries. But again, by carefully dosing the NG2-TD tomato line we use in our presentation, there's no tomato labeling in between 10 micron size to 100 micron size. So that therefore we only focus on vessels smaller than 20 micron size. And that's a critical information to performing any study to visualize and label parasites, and should be very fully considered for your experimental design.
Okay. So yes, that's a good I mean, that's a good point. I have another very quick technical question. So when you subject the mice to hypoxia, what's the age of the mice?
Oh yeah. So the mouse we used is about 8 weeks up to 16 weeks in that age. So the average weight of the mouse we use is about 25 grams.
Okay. 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 Yeah. So is any other questions from otherwise we should thank you for your beautiful presentation, and Dr. Yuan.
Thank you.
All right. Thank you both.
Thank you, Vanessa.
See, we are out of time. And if you have a question, for those of you, please email info@navbo.org, and we'll make sure to share your questions with Dr. Yuan. So I hope you found this research presentation beneficial. These webinars are brought to you by the NAVBO Education Committee. Our next webinar is Thursday, March 12, 2020 at 1 o'clock Eastern Time with Dr. Brent Weinstein from the National Institutes of Health. Look for registration information in the NAVBO Newsbeat newsletter. Please fill out the GoToWebinar evaluation form when you receive it in a subsequent email, and let us know what you think of the GoToWebinar format and any topics you may want to see in a future NAVBO webinar. Thank you very much, and until next